Well, this year's conference, like most of the WIN symposia, I think really has brought out some of the most innovative speakers, the most innovative science. There's always something new at the WIN symposium that I hadn't heard somewhere before. This year, I think we had a fabulous talk by Andrea Califano, who always gives a terrific talk, but building on the systems biology approach that he's been developing to identify master regulators, switches in molecular pathways that, if targeted, have the potential to be much more effective as targets than the traditional sort of single gene, single protein targets that we've been using up until now. I thought the concept that was presented of viral mimicry was also fascinating. The concept being that we are programmed through evolution to have immune systems that are very effective in fighting viruses, because viruses are infections that babies get. And so if you don't have a good immune system to fight off a viral infection, you're not going to survive childhood. Cancer is a disease that mostly older people get. And so the immune system is not as well prepared to fight off cancer. But if you can somehow make your cancer look like it's a cell that's been infected by a virus, then all of a sudden the immune system potentially becomes much more effective in targeting those cancer cells. So I thought that was a really interesting talk. The remarkable things about immunotherapy, as we've now seen it unfold, are that it seems to work in just about every cancer type. And there's always a proportion of patients who have a long-term remission. And so our challenge is to elevate that proportion of patients who have the long-term remission. I think as we are beginning to learn now which patients are the most susceptible to immunotherapy, and there are various biomarkers that have been in development that seem to be able to identify patients who are most susceptible. But the one that's becoming very compelling is a measure of the total mutational burden in the tumor. Again, the concept being that the more mutations, the more abnormal proteins are produced by those mutant genes, and those abnormal proteins then become recognizable by the immune system. So then if you take the brakes off the immune system with a drug like an immune checkpoint inhibitor, then you really, the immune system can really seek out and destroy the cancer cells very efficiently. So it's a very interesting time. Yeah, well, we certainly heard that repetitively throughout the meeting so far, that the great excitement about precision medicine is that when you have a specific target that is a driver gene in the tumor, and you have a good inhibitor of that target, clearly, patients can get very immediate and very profound clinical responses. The problem, of course, that we've all come to recognize is that the tumors have a remarkable ability to become resistant, so those responses are not very long-lasting. So, the concept that's been developed in all of medical therapeutics, from tuberculosis to Hodgkin's disease to HIV, is combination treatment. Hit the offending agent, whether it's a tumor cell or a virus, with as many drugs that hit as many vulnerabilities as possible. The big challenge, one of the big challenges, of course, is the tolerability to the patient of receiving those drugs. Now, the good news is, many cancer drugs are far less toxic than the chemotherapy drugs of years gone by. So I think it's going to be possible that, again, there's not going to be a single, double, or triple drug combination that's going to work in every person. There may be unique combinations for each person. But this is where I think WIN is really taking a global leadership role in developing triple therapy for lung cancer with the SPRING trial that's now in development for patients with advanced non-small cell lung cancer, where we already now have a pharmaceutical company sponsor who's going to provide 3 drugs to support that study. So that study will certainly go forward. And then similar concepts, being applied now in other settings within the WIN consortium. So it's, I think we're clearly in the forefront there. We had a session yesterday afternoon where we were having a debate really between 2 very well-established investigators as to whether the clinical trial designs that we've relied on for many years, like prospective randomized clinical trials, still have a place in this modern era of targeted therapy for cancer. And Christophe Letourneau presented an update of his analysis of the SHIVA trial, which various people have interpreted in various ways. I personally think that the fact that the SHIVA trial did not demonstrate a benefit for patients receiving the matched therapy is not an indictment of the field of precision medicine. It only is an illustration of how complicated it is and that we need to continue to do research. Now, Dr Kurzrock pointed out some of the flaws in the SHIVA trial and also pointed out, I think rightfully so, that it's going to be very difficult to continue to advance precision medicine solely by doing prospective randomized trials. They take a long time to set up, they take a long time to complete, the field is moving very quickly, and we need different kinds of clinical trial designs to be able to move the field forward. The one thing that we can't do, I don't think, is just move to widespread adoption of precision medicine. We heard, following the formal debate, many comments from people in the audience yesterday, some of whom just said, well, we're ready to just move straight into giving everybody a precision medicine approach without any proof that that necessarily is of value, and importantly, without any mechanism to capture and record the outcomes of those patients so all the rest of us can learn from those experiences. I think that's going to be critical going forward. Well, I'm looking forward actually to the session immediately following lunch today, in which I will be participating, which will be sort of a roundtable discussion among many experts in the field, but focusing on what do we really need to do to continue to accelerate the development of precision medicine approaches. I think there are many things we need to do. Personally, I think one of the most important ones is to develop a common approach to annotation of all the genomic variants that are found in patients' tumors. The tumors are remarkably heterogeneous and complex. We oftentimes don't understand the biological or clinical significance of the abnormalities found in the tumors, and we can't act on those abnormalities if we don't know what they mean. And we need sort of a common approach with common standards, common vocabulary, common definitions to be able to collectively provide the annotation that's necessary to really apply precision medicine to its full potential. Absolutely. It's been very much that way, and different laboratories report their results in different formats, different content. They have different bioinformatics pipelines. They have different strategies that they use for calling out what's a variant. And most importantly, many of the variants that are discovered in patients' tumors are not currently understandable in terms of, there's the so-called variants of unknown significance. The only way we're going to move from having a variant of unknown significance to a variant whose significance is known is, to capture that information in a standardized format so everybody knows what we're talking about, and then capture information about what happens to the patient and what their outcomes are so we can understand what are the consequences of that variant being present. As I said at the beginning, I think the WIN Symposium is one of my favorite meetings. I go to lots of meetings. We all go to lots of meetings. We hear some of the most cutting-edge science at WIN delivered by people who are working, who are the leaders in the field. There's ample time for networking and discussion. I think we'll continue to to see this very forward-looking view of precision medicine being described at the WIN Symposia going forward.